So good morning. It's a real pleasure to be here to tell you about the human sequence variation data that we have in neXtProt. I hope you are seeing my title slide. I'll begin with a very brief one-slide introduction to neXtProt, and I'll go on with the sequence variation data to give you an idea of the corpus of the data that we have. I'll then go on to tell you how you can access the data, and then I'll wrap up. So to begin with, what is neXtProt? So it's a knowledge base on human proteins, which is funded by the Swiss ELIXIR node, the SIB, Swiss Institute of Bioinformatics. We're an integrative knowledge base, so we take data from a number of different data sources and present it to the user in a platform that should enable them to explore the universe of human proteins. And maybe just as an aside, we're involved in the proteomics community. We've been working with HUPO, the Human Proteome Organization, for a number of years. So you saw probably neXtProt as one of the resources in Juan's slide. And we're also active in the EJP, the European Rare Disease Project. And so that's sort of the introduction to why I'm going to tell you about variants today. So let's now take a look at the global picture of what the data is that we have. So we have data from polymorphisms from normal subjects who have not been diagnosed with a disease, and we have variants from patients. In terms of the polymorphisms, we have data from dbSNP, and we also integrated data from Nomad, so we have the allele frequency data. In terms of the variants that are associated with disease, we have variants from COSMIC, which are obviously variants that have been seen in cancer patients. We have variants that we've annotated from the literature, either for genes that are involved in cancer or for genes involved in channelopathies. And we also have all the disease-associated mutations from UniProt, which obviously deal with genetic diseases. So that's in terms of the variety of the data. So we have data from 5 sources. If I give you now the big picture for what is currently in our data release, we have over 19,000 entries that have at least one variant, so that's about a 96% coverage. We have over 9 million variants if we include the disease mutations. We have a large majority, 94% of them, which are non-synonymous single amino acid variants, obviously in protein coding regions. Concerning the diseases that are related to these variants, we have over 4,000 genetic diseases and 810 different cancers associated to the variants. So that's the corpus of the sequence variation data that we have so far. Let me now walk you through how you can access the data and what kinds of things you can do. So I'm going to take you through, first of all, how you find variants in neXtProt, assuming that you have an identifier. So in this example, I've taken the same variant from MSH6, P2S at position 42 of the main isoform, and you can either search for a COSMIC ID, a dbSNP ID, a Nomad ID. You search in the proteins and you include silver, and you will retrieve the MSH6 entry, and then you can go on and look at the entry views to get more detail about the variants. So should you not find a result with an entry, with an identifier, it's possible that we are out of sync with the source. So take a look at the entry and see if it's there, but maybe breast cancer susceptibility protein. By selecting the variant in a variant thread, we also show the 3D structures from PDB, and that by selecting a variant, so I've selected the variant we talked about before from BRCA1, we see it highlighted in the structure, and we see that in fact it lies at the C terminal of the first alpha helix in BRCA1. So in this way, you can see where your variant is located and get a feel for maybe does it have an effect or not. Finally, there's the phenotypes view, in which we see all of the possible effects that we've annotated for variants in the proteins. So in this case, in BRCA1, there are a total of 63 phenotypes that we annotated. The summary is shown here. So you see that there are variants that have or may not have an impact on the molecular function, on a biological process, where they're located in the cell, an interaction with another protein, a property of the protein, or a mammalian phenotype. And by scrolling down, you will find a variant and see, does it have an impact or not? And if it has an impact, is it mild, moderate, or severe? So that's the phenotypes view. All of the data that we're showing in our different views is accessible from our API. So it's all tied back to an entry. So you select entry, you then select variant, and you can export all the variant data in either JSON or XML format. And you'll see there's a single URL to do that. Of course, I've shown you how to visualize the data in our interface, how you can access it with the API, but maybe you want to do more. Maybe you want to answer complex questions involving variants. And to do that, you can use the advanced search, which involves Sparkle. So you select it at the top of the search box. We've written a number of different Sparkle examples, so you can select all those examples that have the tag variant and look and see those examples, or you can obviously write your own query in the query box. And just to give you a feel for the types of questions that you can answer with Sparkle that you can't answer with just a simple text search are things like find me all the proteins with at least one variant of the types A to R or R to A, or find me all the proteins located on chromosome 2 and having at least one variant in phosphorylated tyrosine. So you see, you can be exquisitely precise about what criteria you want to use in your queries. So the advanced search searches the data in neXtProt, but you can in fact go beyond neXtProt through the use of federated queries. In this case, you federate your query with other resources that have a Sparkle endpoint. And to give you a taste of that, I've showed you an example where of a federated query between neXtProt and DisGenet, where you can retrieve proteins involved in disease due to intronic variants with one selected publication. And obviously, this would not be possible with the data that's only in neXtProt because we have protein coding variants, but it is possible through federated queries. And you'll note there's a tag federated queries where you can find additional examples. So to summarize, I've told you what is in the corpus of data that we have of over 9 million variants, human sequence variants. I've told you you can search to find variants using the COSMIC, dbSNP, and GNOMAD identifiers. Using the different entry views, you can see what the variation associated with the variant is, its frequency, where is it located, does it lie in a domain or not. You can see what effect it has in terms of the phenotype. All of this data is accessible through the API. You can query it using Sparkle, and I didn't mention, but you can also download it from the files in our FTP site, for instance, in XML format. And all of this data is reusable. It's available under a CC by 4.0 license. So, of course, all of this is a team effort. I'm just the one presenting here. So the neXtProt team is led by Amos Bayrak and Liddy Lane. We have one biocurator, Paula. We have 3 developers, Pierre-André, Alain, and Valentine. We have 3 researchers who are also developers, Mathieu, Kazoun, and Vimel. And I look after the quality assurance. So I invite you to take a look at our website, see if there's any data that's missing, see if there are things you'd like to do that you can't, and let us know, of course. And you can follow us on Twitter or also on ResearchGate. Thank you very much.